Part 3. Growing Mycelia and Mushrooms. Key to growing mushrooms, whether for deploying myco-restoration strategies or eating or using them medicinally, is to first grow mycelia, the subject of the final part of this book. Mycelium is the cellular fabric of our food web, and although pervasive in nature, getting a mycelial mat to infuse through a virgin habitat is both an art and a science. As cultivators, it is our job to help mycelia navigate through the highly competitive microbial universe, one habitat at a time, always keeping hungry parasites at bay. Thankfully, all of the challenges that come up when growing mycelium, even those early hurdles, choice of strains, how to get mushrooms into culture, and how and where to project the mushrooms, can be overcome using the techniques described in the following chapters. Most important of the techniques to come is how to use natural spawn, that is, how to transplant and nurture wild spawn for myco-restoration projects. Wild spawn has the major advantage of being already acclimated to habitats teeming with competitors. Conversely, pure culture spawn, bought from commercial spawn suppliers, must either be implanted into clean substrates or slowly adapted into the complex microbial ecosphere that surrounds us. Natural spawn can be made by transplanting wild patches of mycelium from germinating mushroom spores and from regrowing stem butts. For many, buying and using pure culture commercial spawn can speed up the early steps of the process, but the mycelium ultimately fares better if it is inherently fortified against the onslaught of wild microbes eager to consume it. In the end, taking the time to create natural spawn will prevent a lot of very time-consuming troubleshooting that goes hand in hand with starting from pure culture spawn. For the stubborn pure culture spawn devotees, however, there exists a compromise. Commercial spawn can be naturalized through a process analogous to vaccines. It can be brought into contact with less than virulent dose of the microbes found in the destined habitat. The mycelium's natural antibiotics pre-select microbes that help it survive in the wild. A synergistic blend of microbes joining with the mushroom mycelium provides a defensive shield, much like fortifying an immune system, that not only forestalls competition, but streams nutrients and mycelial stimulants. Once the pace of mycelial growth quickens, regardless of its beginnings, mats can become quite large. In the extreme, they can cover thousands of acres. Remember our myco motto, move it or lose it. A mycelium grower is really a mycelial herdsman. No matter how successful you are in getting mycelium to adapt and grow in one habitat, that success is a temporary episode in the theater of life. Mycelium consumes its preferred habitat resources and then strategizes for transporting itself to new niches. Unless the mycelium is recharged with basic nutrients, it will move on as it transforms debris fields into soil. Mycelium is, in essence, a digestive cellular membrane, a fusion between a stomach and a brain, a nutritional and informational sharing network. It is an archetype of matter and life. Our universe is based upon these networking structures. Your job is to become embedded into the mindset of this matrix and use its connections for running with mycelium. Chapter 9. Inoculation Methods. Spores, spawn, and stem butts. This chapter describes methods of inoculating by using spores, spawn, and stem butts. Although each method can generate mycelium and eventually mushrooms, my preferred method for outdoor colonization is to combine uses of spores, spawn, and stem butts, techniques I will explain as I progress through the chapter. The wisest method for generating mycelium is the one that works. My experiences of success and failure may differ from the experiences of someone from India, for instance. Climate makes a huge difference, as do the seasons.
Some thoughts on failing. Over the years, I have learned the value of attempting that which seems unlikely to succeed. Several times I thought I had failed, only to have the mycelium surge and fruit later. My failures oftentimes become my successes, but with every failure, if I have paid attention, I hone my skills and sensitivity to the mycelium's needs. Hence, another one of my mottos: every failure is the price of tuition I have paid to learn a new lesson. Your method of inoculation will depend upon your access to spawn or mushrooms. Buying pure culture spawn has its advantages, but in this chapter, which could be titled Liberation Mycology, I'll show you how to use nature as your source for mycelium. The materials upon which mycelium is grown or inoculated are called substrates by mushroom growers. Substrates usable for mushrooms are extraordinarily varied, although most come from byproducts from forestry and agricultural practices. Several factors drive the decision-making process. The habitat you want to inoculate predetermines which mushrooms are suitable. Your ability to obtain mycelium in the form of spores or commercial spawn, or by gathering naturally growing mycelium, also influences your decisions. Other factors: experience, labor, ease, and commercial or recreational intent come into play. Because so many methods are now available, you have many alternatives to choose from. I've learned to make the wisest choices by listening to the mycelium. Musings on using spores, the fairy dust of mushrooms. Frank Herbert, the well-known author of the Dune books, told me his technique for using spores. While I met him in the early 1980s, Frank enjoyed collecting mushrooms on his property near Port Townsend, Washington. An avid mushroom collector, he felt that throwing his less than perfect wild chanterelles into the garbage or compost didn't make sense. Instead, he would put a few feathered chanterelles in a 5-gallon bucket of water, add some salt, and then after 1 or 2 days, pour this spore mass slurry on the ground at the base of newly planted firs. When he told me chanterelles were growing from trees not even 10 years old, I couldn't believe it. No one had previously reported chanterelles arising near such young trees, nor had anyone reported them growing as a result of using this method. Of course, it did work for Frank, who was simply following nature's lead. Frank's discovery has now been confirmed in the mushrooms industry. It is now known that it's possible to grow many mushrooms using spore slurries from elder mushrooms. Many variables come into play, but in a sense, this method is just a variation of what happens when it rains. Water dilutes spores from mushrooms and carries them to new environments. Our responsibility is to make that path easier. Such is the way of nature. Frank went on to tell me that much of the premise of Dune, the magic spice spores that allowed the bending of space, tripping, the giant worms, maggots digesting mushrooms, the eyes of the freeman, the cerulean blue of psilocybe mushrooms, the mysticism of the female spiritual warriors, the Bene Gesserits influenced by tales of Maria Sabina and the sacred mushroom cults of Mexico, came from this perception of the fungal life cycle, and his imagination was stimulated through his experiences with the use of mushrooms. Spores: how mushrooms travel distances. Mushrooms reproduce through spores that can travel great distances via water, by air, or by hitchhiking on other carriers. For instance, many species of mushrooms use insects to carry spores. The scent of the birch polypore, Piptoporus betulinus, attracts beetles that then burrow into the mushrooms, piercing the spore-rich underlayer to feast on the soft internal flesh. In doing so, the beetles become covered with spores. When the beetle travels to another tree and burrows into its bark to lay eggs, the tree is inoculated with spores of the birch polypore. The emerging fungus provides the beetle's developing larvae with food. The tree softens as mycelium grows into it, and soon woodpeckers arrive in search of beetle grubs and other insects that are attracted to the myceliated wood. Once the woodpeckers leave, carrying spores with them, other birds and insects take up residence. An entire ecosystem spirals from the pock-marked tree, which can become a launching platform for more fungi, insect, and birds. Many other polypore mushrooms coexist within such a tree. And not only do beetles and birds spread mushroom spores, but bears do too. I have seen scratching on trees in the Hope River Valley in Washington state, where bears scratch trees to mark their territories and in the process create apt habitats for spore entry. Mycelium and mushrooms sprout from these scratchlings, attracting more beetles and bugs, and the process begins again. These intersecting cycles sustain biodiversity. Bears, birds, insects, and animals are not the only carriers of spores. Humans carry them too. Many mushroom species turn up on my property after I carry specimens home from forays. When I was studying the taxonomy of psilocybin-containing mushrooms at the Evergreen State College in the 1970s, I was amused to find that many of the species I collected from miles away began appearing in the wood chips around the laboratory. I soon realized that boots, bikes, books, and backpacks are convenient vehicles for spore transport. Millions of opportunistic spores trail like fairy dust behind us. Airplane travel and mail service spread spores throughout the world. Even handshakes spread spores. We are attracted to mushrooms for the myriad flavors and uses, and through our use of them, we spread their spores. Chopping wood, trimming trees, gardening, cutting grass, playing with your dogs, frolicking with your kids, and cooking gourmet mushrooms. All of these activities make us effective vectors, allies, unwittingly recruited by the mushrooms. Whenever you handle mushrooms, their spores stick to you. The wind blowing the spore dust off our bodies adds to spore mobility. I guesstimate that in the course of a day, the average human accumulates between 10 and 100 million fungal spores on his or her body and clothes. Close contact with others in densely populated cities allows for more points of distribution, although the ambient mushroom spore load may be less than that of suburbs or forests, whereas the ambient spore load of molds may be greater, depending upon the state of decay in the city's infrastructure. In fact, everyone is a mushroom grower, from Manhattan high-rise dwellers to Pacific Northwest loggers, whether they want to be or not. We are all Johnny Appleseeds, or rather Johnny Mushroom spores, in the service of the fungal kingdom. Once you're aware of how spores work, you can use this knowledge to start your own mushroom gardens. Collecting spores for inoculation. The air is a sea filled with invisible spores of microorganisms. In my backyard, using a high-tech laser particle scanner, I commonly detect concentrations of airborne particles exceeding 3 million per cubic foot. Of these particulates, about a third of them fall within the size range of most fungal spores, which typically measure 1 to 30 microns. Many of these airborne fungal spores compete for the same habitat, which hampers any effort to cultivate a single species. Success is a numbers game. You need to find a strategy that favors your chosen mushrooms and discourages competitors. Several tried and true techniques work for gathering enough spores for successful inoculations. The simplest method is to take a spore print. Spore prints on paper. Spores for spawn or mushroom art. When you place a mushroom onto paper, falling spores collect in delicate patterns, graceful and beautiful. The collection of these invisible spores amasses as a visible dust. You never know how a print will look until you remove the mushrooms that has cast its spore dust. The mushrooms discussed in this book produce spores from gills, pores, teeth, or folds. The garden giant, Stropharia rugoso annulata, and parasol mushroom, Macrolepioda proxera, are gilled mushrooms. Reishi, Ganoderma lucidum, is a polypore mushroom with pores instead of gills for sporulation. Monkey's head, Hericium erinaceus, has long, cascading spines or teeth. Morels, Marcella esculenta, and Marcella angusticeps have deeply convoluted folds. Mushrooms increase in their spore release as they mature from adolescence to adulthood. The length of time of spore release in a mushroom's life cycle varies from species to species. Spores released in the wild are easy to see as a dust on the ground or wood directly beneath the mushrooms. Sometimes, taller mushrooms will cast their spores onto the caps of other nearby mushrooms. Nowhere else in botany is a color a key feature for separating so many genera. Although genera vary so much in classic spore color, every spore print is mandala-like. As art from nature, spore prints unveil one of the hidden mysteries of the mushroom life cycle. And making spore prints is not only fun, it is essential for identification. The classic method for capturing a spore print is to place the mushroom, spore layer down, on a piece of paper. The spores then descend like a fine mist. These same spores can be used for culturing in the laboratory and for growing mushrooms outdoors. Here are the steps for taking a spore print from a grown mushroom, such as a portobello or garden giant. Making a spore print. Caution: wear a dust mask when you're handling masses of spores. Anyone whose immune system may be compromised, is allergic to fungus, or suffers from lung disease should be especially careful. Wash your hands afterward. 1. Go mushroom hunting. Before going on a foray, learn about the species you want to grow by enrolling in a class or picking up a field guide. Being a West Coast mycophile, my two favorite field guides are David Aurora's All That the Rain Promises and More, and Mushrooms Demystified. On your foray, choose a mushroom specimen, preferably one that is approaching maturity. With many gilled mushrooms, this means that the mushroom cap is convex in shape but not flat. Once mushrooms mature to a flattened state, spore production usually decreases, and in some cases, the edges will upturn at full maturity. If you prefer, order a mushroom growing kit from a mail order company like mine, Fungi Perfecti. An oyster mushroom kit weighing 8 pounds will produce around 1 to 2 pounds of mushrooms and correspondingly 1 to 2 grams of spores, approximately 1 to 2 billion. 2. If the gill color is not white, use a white sheet of paper. Typing or photocopy paper works well. If the gill color is white, as is the case with shiitake or oyster mushrooms, use a colored piece of paper. 3. Sever the cap from the stem and place the cap, gills, or pores down on the piece of paper. Caps of larger mushrooms do not need to be covered. Smaller mushrooms may dry out, hindering spore release, so it is best to cover with a cup or bowl to lessen the rate of evaporation. 4. Allow the sporulating mushroom to sit for at least 12 hours. When you carefully lift up the cap, you will discover the spore pattern that has collected on the paper. The spores fall in a pattern mirroring that of the gills or pores, and, in the case of gilled mushrooms, pile up in the form of ridges. You can use these for cultivation or as decorative art. If you wish to keep your spore prints as artwork, spray them with the same aerosol fixative that artists use to protect chalk drawings. After being sprayed, these fun-to-make nature portraits are unusable for cultivation and are not allergenic. With most species, you can create additional prints by moving the cap to a clean space on the paper and waiting for another 12 hours or so. Some caps will yield up to 6 distinct prints over a week before the mushrooms dry up or succumb to bacteria, other mold, fungi, or maggots. If the mushroom seems wet, replace the paper daily to remove the excess moisture. Or you can place the mushroom on a bottle cap so it stays elevated off the paper. These spore prints tend to be smudgy and less distinct. If you want to grow mushrooms from the spores on the print, then place the dried print in a plastic resealable bag until needed. Write down the date, mushroom species, geographical location where collected, and any other notes. Keeping a specimen for future identification is always advisable, since identification of mushrooms by spores alone is very difficult unless their DNA can be analyzed. Alternatively, you can make a spore print with spores of any color on a clean pane of glass, my favorite method. Once spores have fallen, carefully remove the cap, overlay with another pane of glass, and tape the edges all the way around. The result is a glass-enclosed spore gallery that can be stored at room temperature for years. Storing spores this way allows them to be easily observed without the risk of contamination by extraneous airborne spores, while the glass protects against damaging UV waves. To retrieve the spores for future use, just cut the binding tape, open the spore gallery, and use a razor blade to scrape the spores into a pile. Spore prints made either on paper or glass, once sealed, can also be sent through the mail with few ill effects. Spore prints come in all of the colors of the rainbow. Within most genera of mushrooms, the color is consistent or varies only slightly. For instance, oyster mushrooms have a whitish or sometimes a light lilac color. Most field guides for identification are organized using spore color as a primary distinguishing feature. Among the better field guides are David Aurora's All That the Rain Promises and More, and Mushrooms Demystified, Gary Linkough's The National Audubon Society Field Guide to North American Mushrooms, and Alan and Arlene Bassett's Mushrooms of Northeastern North America. Among European guides, I like Rosemarie Anke's book, 1200 Pilze, which is currently available in German, Edible and Poisonous Mushrooms of the World by Anne Hall, Stephen Stevenson, Peter Buchanan, Wang Yun, and Tony Cole, is a good overall guide. The bag technique of collecting spores. Some mushrooms cannot easily spore print on paper. In particular, morels and lion's manes are architecturally different from classic button-style mushrooms and can broadcast spores by expelling them in all directions. Collect spores from these mushrooms by enclosing them in a paper wax bag or plastic bag. For best results, keep the mushroom upright as it is in nature, tenting it an upside-down bag so insects and other debris will fall out. A paper bag has the advantage of being biodegradable, whereas a plastic bag allows for a cleaner collection surface when you're amassing larger quantities of spores. However, plastic bags sometimes collect so much condensation that spores are rinsed down the sides in an unusable sludge. I prefer paper sacks, especially for morels. The sacks can be sandwiched into cardboard to create cardboard spawn that can then be placed outdoors. Collecting spores from air streams. Air passing over mushrooms carries spores. Studies by E.R. Badham show that psilocybe mushrooms, like many others, orient themselves into the wind, a response known as aerotropism. A pressure drop forms under the cap, allowing spores to be sucked into the ambient air streams. And if you channel air past a sporulating mushroom into a chamber, you can collect many spores. Here are 2 ways of accomplishing this. There are no doubt more, so feel free to improvise. 1. Place a nylon air sock around the exhaust tube coming from a forced air mushroom dryer. As the mushrooms dry, spores from the drying mushrooms flow into the sock. Make sure your dryer is set at the lowest possible heat setting so that drying does not occur too quickly. 2. Use an aquarium pump or a small peristaltic air pump to suck air from the spore collection chamber. With the sporulating mushrooms, gill up and aim the spore-enriched air directly into a paper bag, onto a pane of glass, or into water if you want to make a spore slurry, which in most cases would have to be used within 48 hours or frozen until needed. This method channels spores to a specific target. If the tubes clog with spores, use a probe to free the airflow. Gazillions of spores can be amassed with these methods. If you want to gather spores from thousands of mushrooms, particularly oysters, shiitake, reishi, maitake, and other cultivars, I have an elegantly simple method. Contact your local grower of exotic mushrooms and ask for spores from their air ducts in their growing rooms or from their spore-laden air filters. Most cultivators, small or large, use plastic ductwork and coarse air filters in their ventilation systems. As the air is recirculated, the spores from the mushrooms get sucked into the ducting and collect en masse. The ductwork can be taken down and the spores can be shaken into an airtight gallon jar. Up to a liter of spores can be gathered from 1,000 square foot growing room every few months. Store the spores in a cool, dark place. Using a desiccant during storage will keep your spores dry, preventing premature germination and reduce competitor molds that feed on the nutrient-packed spores. In fact, the spores of most temperate species, after a period of drying, can be frozen for many years. However, I found that the spores of most tropical species, like their Mycelia, die when frozen. Although the vast majority of the spores in the ductwork will be from mushrooms being cultivated in the growing room, bear in mind that spores of outside fungi and contaminant mold fungi will inevitably be in the mix. The cleaner the crop inside and the cleaner the incoming air, the more pure the collected spore mass will be. If more than one species is being grown, then obviously the spore mass will reflect the type of mushroom sporulating in the growing room. Harvesting spores from dried mushrooms. This technique is more difficult than most others listed. Occasionally, I want to start a culture from a dried mushroom specimen. Dried specimens have plenty of spores on their gills, but many times the spores are not easily cultured when in contact with the gill flesh, soon contaminating when dehydrated. Using high heat in the drying process may lessen viability. Nevertheless, to release the spores, allow the gills to float in water, which will rinse the spores off. They will temporarily float before becoming saturated with water and sinking. Before they sink, you can skim the water's surface with a fine sieve to remove the spores. Collecting spores with variable mesh screens and centrifuges. This is a fun technique for gathering spores, especially from the subterranean truffle-like mushrooms, like the mycorrhizal, rhizopogon, and glomus species, which live above the root zones as well as underground. Gather the mushrooms at maturity. Dry brush the outer layer using a piece of mattress foam and allow it to dry, a process that may take hours or days. The spores often mature as a defensive response to being plucked. Pulverize the dried mushrooms in a household food blender and place them on a stack of shaker screens with decreasing mesh fineness from top to bottom. Since most mushroom spores measure between 3 and 20 microns, finer screens will eliminate all but the smallest non-spore particle. Most of the time, 3 or 4 screens will suffice, ideally with the finest screen openings measuring 0.0007 inch or 18 microns. If this type of screen is not available, using a 0.007 inch screen, 180 microns, will work, although some impurities will fall through. The coarsest screen can be of the window variety, but it should be rinsed with water and dried before use. For making ultra-pure samples, place the spore mixture into distilled water and place it in a centrifuge. Since spores have a fairly consistent color and specific gravity, distinct populations will soon settle out and form bands of discolorations. If your mushroom has a distinct spore color, you can remove the undesirable layers with a turkey baster or syringe until you come to the spore stratum. Rinse well before drawing from within the spore-producing layer. Although color will be a good indicator of which strata has the spores you desire, should there be any question, a quick look under a microscope can tell you if the shape and size matches your mushrooms. Please consult one of the previously mentioned recommended mushroom field guides that have good microscopic descriptions. Isolating spores by electrostatic fields. Mushroom spores tend to be electrically charged and so adhere to opposing charged surfaces. A coating of spores is most noticed on the walls in a room or on other dry, well-grounded, positively charged materials. Electrostatic filters, widely used in homes, can be set up downwind of a mushroom kit to collect 99% of the spores produced from the mushrooms. Before trapping the spores, thoroughly clean the filter screens. After the filter is unplugged, the screen can be cleaned to collect the spores using a brush, or better yet, pressurized air from, for instance, an air compressor. Blow the spores into a suitable vessel, such as a large plastic or paper bag. Wear a mask so you don't inhale spores. How many spores can a mushroom produce? Oyster mushrooms have been reported to convert 50% of their mass into spores, a figure that seems high to me. Nevertheless, if this were true, a pound of fresh oyster mushrooms from a single mushroom kit would yield about 45 grams of dry mushrooms. If the maturing mushroom released 50% of its mass in spores, or 22.5 grams, then the output would be approximately 25 billion spores. Even if this estimate is off by a factor of 10, in other words, if a kit produces only 2.3 grams or 2.5 billion, a more realistic number, since there are an estimated 1 billion spores per gram, measuring about 10 microns in length, then they would span 25,000 meters or 15.5 miles if laid end to end. Perennial mushrooms produce many more spores than annual species like oysters. Aurora estimated that a large artist conch can produce 5 trillion spores annually. With spores averaging 8 microns in length, this means that a string of these spores would be 40 trillion microns, or 40,000 kilometers, or approximately 24,854 miles, just about enough to encircle the Earth. This abundance of sporulation of just one species, when placed in the context of total fungal biodiversity, shows how infused our biome is with fungal DNA. Mushrooms are mycological geysers spewing spores by the billions and competing with each other to dominate an ecological niche. A new astromycological unit, the amount of spores equivalent to encircle the Earth, the spore Earth unit, could be used, for instance, to calculate the outflow of fungal spores into space. The Earth is sporulating, seeding the galaxy with its germplasm. How many SCUs are leaving the Earth's atmosphere every day? Traditionally, scientists have thought about panspermia from outer space that could infect the Earth. Equally likely, the Earth is inoculating the heavens. Germinating spores. With most saprophytic mushroom species, germinating spores is not difficult. Spore germination depends upon the species, the material upon which they are grown, and the temperature. Each species reacts differently to different substrates. The genetic variability in spores allows for options in growth media and methods and is part of fungi's adaptive strength. This is nature's way of guaranteeing a mushroom's survival should its natural habitat suddenly change. Some mushrooms are highly adaptive to a wide range of substrates. Oysters, turkey tails, and Hypholoma fungi are amazingly aggressive. Their spores germinate with ease. On the other hand, in my experience, shiitake spores must dry first and then rehydrate before they can germinate en masse. Spores may not germinate for any number of reasons. One problem with spore prints is that when the protein-rich spores concentrate, they become a fertile breeding ground for bacteria. Spores need to be diluted in order to tip the balance in favor of fungal growth and against competitors. In the laboratory, the common practice is to germinate spores under sterile conditions on nutrient-enriched agar media. See Growing Gourmet Medicinal Mushrooms and the Mushroom Cultivator. Most mushroom spores love to grow on the moist surfaces of dead plants, especially when they're scattered like checkers on a checkerboard. Spores germinate quickly in water. The problem is that most life on this planet also loves water. So when you immerse spores in water, other organisms, especially bacteria and protozoa, consume the fungi as food. The art of cultivation is to give the mushroom spores a head start, in advance of competition to initiate the ever-increasing spiral of germination. Ideal spore dispersal is a balance between the conduciveness of the habitat and its background population of competitors. Once a few spores germinate, adjacent spores are triggered into germination. The following descriptions outline a variety of successful techniques. You'll discover the best one for you and your mushroom through trial and error. Spore germination in nutrified water, a sugar and salt broth. In most instances, fresh spores of saprophytic mushrooms contain enough nutrition to germinate in water without added nutrients. However, as they get older, age becomes a barrier to germination. Soaking spores in a sugar and salt broth often causes them to germinate more quickly than competitor spores. After 1 to 2 days, the actively germinating spore mass slurry will be ready to transfer, so be ready. If you incubate it too long, the decline in spore viability will provide an opportunity for hostile competitors to grow and eclipse your chosen mushroom spores. The activity in this broth is a microbial race, and your coaching goes far toward determining who wins. Spore prints from paper and glass are the easiest to use with this germination method. By scraping the spores off the glass or paper with a razor blade, you can collect about 0.5 to 1 gram of spores, approximately 500 million to 1 billion spores, for use in your solution. The salt limits bacteria growth without stifling spore germination. To make a sugar and salt broth, 1. Add 1/4 teaspoon non-iodized salt and 1 tablespoon sugar or light molasses to 1 gallon of water in a large pot and boil for 10 minutes. Remove from the heat. 2. As it cools, pour the broth into a clean, well-rinsed container made of plastic, stainless steel, or glass. Make sure the container has not held chemical products and/or milk. 3. Once the broth is cooled to room temperature, add 1 teaspoon 1 gram or 1/28 ounce of spores. Use a dust mask and do not inhale while working with the spores. Wash your hands thoroughly. Avoid contact with spores if you have fungal allergies. Alternatively, you can use the water left over from immersing 2 or 3 mature 4 to 8 inch wide mushrooms, for example. However, spore broths made from spore prints tend to have fewer bacteria than those made from spores from submerged mushrooms, or you can rinse spores off a mushroom's gills, allowing the spore concentrate to collect in a gallon of water. Never use soaring mushrooms. Use only those that are pristine, firm, but fairly mature, and growing rapidly. Many mushrooms continue to grow after we pick them, as their flesh is transformed into spore germinating material in a last heroic effort to produce offspring. 4. Cover the broth immediately and incubate it for 24 to 48 hours in a shaded place with even temperatures between 50 and 80 degrees Fahrenheit. Temperature preferences are species specific. To zero in on the best temperature for germination, check a field guide that identifies the range of temperatures at which the mushroom fruits in the wild. Often but not always, this will be in the temperature window for germination. 5. During incubation, shake the broth vigorously twice a day. Depending upon the species and circumstances, a hyperactive state of germination is triggered and fine threads of mycelium, barely visible, float in the media. If the spores are old, fewer will germinate, which may mean incubation in water should be extended, but not more than 5 days. Otherwise, bacteria growth may overwhelm spore growth. Generally, you should use spores not more than 2 years old. You have just created a gallon of spore broth, a liquid spore mass slurry, useful for inoculations. Once the spores have sat in the water for 1 or 2 days, they can be broadcast onto a substrate. This is the simplest method of cultivation, but it is not the most successful in every circumstance. Whether you are successful in growing mycelium and then mushrooms depends upon many factors, some obvious and some mysterious. Using the liquid spore mass slurry for inoculation. When spores are collected under sterile conditions and then put into a sterilized nutrient slurry on a laboratory stir plate, a more technically demanding technique than the simple technique described above, spore germination usually occurs in about 3 to 4 days. By day 10, the mycelium is so thick it collects on the top in the form of a mat, the size of a pancake. In nature, spores face more challenges, especially from other microbes. To limit the challenges to your spores, choose the cleanest and freshest possible substrate. If the substrate houses smaller populations of microbial competitors, and if enough spore mass is placed on the substrate, your attempts have a better chance of succeeding. Spore mass slurries can be used for inoculating coarse substrates like straw, wood chips, grasses, and human-made materials like pellets for wood stoves, kitty litter, rugs, textiles, and cardboard. Spores have the advantage of quantity, many million points of inoculation. Their disadvantage is their fickleness. Sometimes they work well and other times not, depending on the species and the substrate. There are other ways of using spores. Spores in oils. Spores can be immersed in canola, corn, or safflower oil, which can be used as a lubricant for chainsaws or other cutting equipment. As trees, brush, or plants are cut, the spore-infused oil distributes spores to the newly cut surfaces, an efficient method of transfer. Another advantage of using oils is that they help the spores stick to their surfaces upon contact and have less chance of being washed or blown away. I filed a patent on spores in chainsaw oil, but another inventor from Europe had already come up with a similar idea first. His idea was to introduce a benign fungus that would compete with the parasitic mold that infected shrubs after cutting, so the patent office rejected my patent application, but I am reapplying in the hope of getting it approved, as my approach is to speed up decomposition and habitat restoration, a different but complementary goal to preventing competition. We did a simple test using 10 million, 100 million, and 1 billion spores of oyster mushrooms in 1,000 milliliters of canola oil. With the 100 million spores in the liter, we had the best results in getting good mycelial growth using an estimated 10 milliliter of spored oils when cutting through an alder tree, about 6 inches in diameter. The solution of 10 million spores also did well, but it took another 2 weeks to catch up to wood slices that were inoculated with the richer 100 million spores per liter. I am sure that lesser concentrations would be effective, but probably would require additional time to achieve the same mycelial mat density. We plan to conduct more tests in the future. The solution of 1 billion spores did not do well because when spores are too concentrated in one space, they become a fertile substratum for bacteria before they can germinate and mobilize a defense. Germinating spores on cardboard. Germinating spores on corrugated cardboard is a good method for creating cultures. Corrugated cardboard, with its ridges and valleys, favorably selects mushroom spores to the disadvantage of many other fungi. The sweet wood-based glues used in cardboard provide a boost to mycelial growth. I really do not know why, but contaminating green molds, like Trichoderma species, do not grow as well on cardboard as many gourmet and medicinal mushrooms do. When you tear apart a box and soak a 1 by 2 foot piece of cardboard in hot water for 1 hour, the fibers soften, making a friendly bed for spore germination. Place a mushroom upon the moistened cardboard and let it sit until its pores have fallen en masse, usually overnight. Remove the mushroom and incubate the spores on the cardboard for a period of a few weeks in a container set in a cool, dark place. I prefer plastic storage containers with snap-on lids. After 1 week and periodically thereafter, open the storage container to inspect for germination. The cardboard should remain moist. Usually, a small volume of excess water will remain on the bottom of the container and will keep the humidity high and the cardboard continuously moist. Once colonies are visible, transfer this mother colony of cardboard to a sheet of soaked, corrugated cardboard about 4 times as large for further expansion. This can be done repeatedly every 2 to 3 months until you have enough cardboard spawn for starting a mushroom patch. Germinating spores on straw. One method that is useful to those living in colder climates is the transfer of spores onto moist, untreated straw. Here I use the term colder loosely, meaning when temperatures hover between 35 to 55 degrees Fahrenheit. Spores can be gathered from a spore print on glass or from a spore sock, or by other methods you prefer. Immerse 0.1 gram of spores in a liter of water and then spread the mixture over 10 pounds of straw. Once inoculated with spores, the straw is placed in a suitable container, a perforated bag, burlap sack, cardboard box, or wooden crate, and thereby protected from rapid water loss. In the Pacific Northwest, wet straw does not become contaminated with mold fungi for several months when stored at low temperatures. We have found that if oyster mushroom spores are placed on moist, untreated wheat straw and left outside at temperatures of 35 to 50 degrees Fahrenheit in January, mushrooms begin fruiting in late March. I call this method cold incubation. Although the process is slower than pasteurization methods, it works with little effort, underscoring the mass generated from the new fruitings provides exponentially more mass than the starting spores. What this means is that under certain conditions, cool temperatures, the right substrate, and an aggressive species like those in the genus Pleurotus, the spiral of generation of mycelium mass is potentially infinite. Given the ability of Pleurotus species to break down toxic wastes, this method is one even rural communities without electricity can put into practice. As with all low-tech methods, every attempt will not be successful, but your experiences can be used to hone your skills over time. Germinating spores on burlap. Like the spores of many other similarly aggressive saprophytes, oyster mushroom spores germinate easily on fabric cloth, especially burlap. Choose burlap, usually made from jute or hemp that has not been treated with fungicides, preservatives, or insecticides. Burlap imported from Southeast Asia often smells of kerosene or diesel, used as an insect repellent, but the samples we have received from Canadian and U.S. burlap manufacturers appear to be free of preservatives. This technique can be refined as you learn the preferences of each mushroom. Soak a few burlap sacks in water for an hour or more and then spread them across a flat surface, such as a picnic bench. Place a 4 to 6 inch maturing mushroom for every square foot on the burlap fabric. Cover with newspaper or cardboard to prevent other airborne spores from raining down upon the exposed burlap, to protect the mushrooms from wind, and to prevent dehydration. Allow the spores to drop overnight. Once the spores are discharged, remove the mushrooms. The burlap can be rolled and tied with twine, each sack separately, and stuffed into cardboard mailing tubes. Pour on a gallon or so of water to thoroughly soak the spore-infused burlap sack. To further offset evaporation, put those moistened burlap stuffed tubes into a cardboard box and store the box in a shady location. The gradual release of water will help limit competitors and give the spores a head start. After 2 to 4 months of incubation, remove the burlap from the tubes, unroll and inspect for signs of growth. Re-roll the burlap and return it to the tube if growth is not thorough. Wait another month, then re-inspect. After several months, depending upon the species, rate of growth, and numerous other factors, mycelium will appear as island colonies. The scent of the mycelium, the species' signature fragrance, will be a key feature, allowing you to recognize whether or not the mycelium is resident. Oyster mushrooms, Pleurotus species, for example, emit a unique fragrance that smells mildly but pleasingly of anise. Without the use of a microscope or experience handling the mycelium of the species you want to propagate, you may not know if you have mushroom mycelium until fruitings occur. Your only choice then is to run with the mycelium you see. Once the burlap is at least 25% colonized, meaning a fourth of the burlap is fuzzy white with mycelium, gently unroll the sacks. Stuff the sacks with wood chips, preferably wood chips from a species of tree on which this mushroom species naturally grows. They will look like sandbags. Stack the stuffed burlap sacks outside, 3 high, preferably atop plywood, a pallet, or thick cardboard in a shady location out of the wind. Cover the shade cloth and incubate for several months. The burlap bags, with luck and a little help from nature, will begin producing mushrooms with the onset of conducive weather conditions, usually during the rainy months in the spring and fall. Spawn. Spores germinate into mycelium. When this mycelium is used to inoculate more material, it is called spawn. Spawn can be sourced from the wild or from spawn laboratories. Wild spawn will not be as clean as pure culture spawn, but wild spawn has the advantage of having been in contact with microbes and so may be better able to acclimate to wild habitats. Spawn can come in many forms. The most common forms of commercial spawn are made with sterilized grain, sawdust, dowels, or plugs, or wood chips. Methods for making spawn are described extensively in my earlier books, Growing Gourmet and Medicinal Mushrooms, and The Mushroom Cultivator, co-authored with Jeff Chilton. I like to use laboratory-grown spawn to first create mother patches that incubate for 6 months to a year outdoors, acclimating to the fluctuations of weather and the wild microbial world. I then expand the mother patches from 2 to 20 times their mass using the strategies described in this book. If successful in growing mycelium from spores, a similar expansion schedule can be adopted using spore-generated mycelium. The key is to achieve enough mycelial mass to get the job done. For most people, buying spawn from a reputable laboratory is the most direct and quickest method. I recommend buying commercial spawn for those who are impatient or who do not have ready access to mushrooms or natural mycelium. First, let's look at how to gather wild mycelium from nature. Transplanting wild mycelium. Mycelial footprints on the path to mycological paradise. Fans of mycelium race everywhere. You can easily find mycelium beneath a down log, in among stacks of aging firewood, or in piles of leaves or wood chips. Practically any fallen tree or piece of wood that has been lying on the ground for a few months will host mycelium on its underside. The mycelium pulps the wood over time, slowly digesting its primary components, lignin and cellulose. Identifying mycelium without its mushroom is difficult since it has few characteristics to help you identify it. Some examples that are easily identifiable are the bluet, Lepista nuda, and the garden giant, Stropharia rugoso annulata. The bluet's brilliant purplish mycelium grows underneath some of my fir trees. The garden's giant mycelium has a ropey quality and a rich and uniquely sweet fragrance, which it also emits when grown in the laboratory. In a sense, the mycelium breathes, emitting a fragrance that is a valuable feature to the experienced mushroom gatherer. The outgassing from the mycelium carries scents that are often species specific and recognizable. While walking through the woods, I have often detected the anise rich fragrance of Clitocybodora or the pine like fragrance of Matsutake. My nose knows the smell. Memories resurface and my brain is titillated by the encounter. I then begin to search for the mushrooms of those species, and if I don't find them that day, I make a mental note to check the site again during the mushroom season. Mushroom scents, even in the absence of mushrooms, inform us that the mycelium is alive and thriving. Though in most cases, scent alone cannot be relied upon as an identification tool. You need the mushroom. Mushroom scents can reveal the presence of otherwise hidden mycelial mats. Once you find a natural patch of mycelium, which I prefer to call a mycelial lens, or if it is exceptionally large, a mother lode, you have a golden opportunity to dance through life with a fungal friend. Here is how. The simplest way of transplanting mycelium is to scoop it up and move it to a new location. The transplanted mycelium, virgin spawn, will be used to make a mycelial footprint at another location. When virgin spawn is transplanted and placed into contact with the right mixture of materials, the mycelium will regrow, expanding the colony. Like all living beings, each individual will have preferences for different combinations of food. When providing materials for the mycelium to consume, the obvious choices are to try to replicate the native ecosystem from which it grew, or to find new materials for the mycelium to sample. This task is simpler with the saprophytic mushrooms than with the mycorrhizal ones. This book concentrates on these easier to grow decomposers. Check Chapter 14 for species individual preferences. Many early cultivators simply gathered mycelium-rich logs, having, for instance, shiitake mushrooms fruiting wildly upon them, and then placed those fruiting logs adjacent to newly cut logs. Hosting competitor fungi are removed before the competing mushrooms can sporulate. The developing shiitake mushrooms then cast spore dust onto the new logs, effectively inoculating them. This method can be repeated for many years, but the population must be constantly culled so the selected mushroom predominates. Ultimately, however, other mushroom competitors are likely to take hold with turkey tails, Trametes versicolor being the prime, if not the most benign competitor. Turkey tails are empowered with excellent medicinal, mycoforestry, and mycoremediation properties. Thus, it's a serendipitous quirk of nature that the most aggressive competitor is one of our strongest allies. So if turkey tail is the invader, it should be welcomed and put to use. Saprophytic mushrooms growing in wood chips are some of the easiest to transplant. To find this type of mycelium, look for areas where the chips are held together in a clump. Healthy mycelium grips the wood chips so firmly that some strength is required in order to tear them apart. This tenacity is a reflection of the strength and vigor of the mycelial mat. Once you find mycelium, you need to give it a new friendly environment. Mycelium needs moisture, air, and darkness. A loosely folded cardboard box or paper sack is a fungus-friendly container. Once mycelium is placed into a cardboard box or paper sack, it should be exposed immediately to the outdoors after being transported to your home, unless temperatures are below freezing or too hot. If it is blazing hot, the mycelium is best planted at dusk or early morning before the sun is high in the sky. If you are concerned that it is too hot or cold, the transplanted mycelium can be placed near a cool cement foundation of the house, for instance, in a basement up against the wall. The mycelium can be stored until you are ready to move it outdoors. In this case, I lay another panel of cardboard on top of the natural mycelium as an extra buffer against evaporation. Apply water every 2 to 4 weeks as needed, pouring about 1/2 gallon in a 2 by 3 foot box. Store your transplanted mycelium in the shade if you incubate it outdoors until you prepare your mushroom bed. In some cases, the mycelium can stay healthy for months after leaving its native habitat, especially in cold temperatures, since growth is slowed as temperatures drop. When spring returns, the mycelium can be planted outdoors in your newly prepared mushroom bed. In my experience, I have found that the ideal time for transplantation is from March to June. Mycelial footprints and the wonders of cardboard spawn. One method I favor for expanding naturalized mycelium is to use cardboard. The idea for this method came to me when I observed an elderly couple who were suppressing weed growth in their rhododendron garden by placing cardboard over the mulch around their rhododendrons. Seeing my wife use cardboard in the garden for a similar purpose reinforced the idea. Although the husband and wife team were seeking to minimize weed growth without the use of chemicals, they unexpectedly grew mushrooms. Covering wood chips and mulch with cardboard promoted mushroom growth. Since that initial observation, I have adapted this method, improved upon it, and used it for running mycelium outdoors. The elegance of this method is in its simplicity. Another variation of this method is described on pages 146 and 147. To use this method, first flatten 2 corrugated cardboard boxes. Cardboard from the United States, Canada, and Europe tends to be free of dioxins, thanks to environmental regulations, and is not toxic. Cardboard from other countries may have some residues. Tear the cardboard panels so that the corrugations are exposed. Wet the cardboard until it is saturated. Take the torn panels and place them with the exposed corrugation side in contact with the mycelium recently implanted. You can encourage mycelium to grow upward through the wood chips or mulch and surface just beneath the cardboard. If the mycelium already resides just below the surface, the patch is maturing. Scrape away the top layer of wood chips or mulch to expose the mycelium to the cardboard. The sooner the terrestrial mycelium surfaces and makes contact with the wet cardboard, the better. For this method, I prefer aerial mycelium with uplifting fans of growth. If the air is dry, first saturate the bed of mycelium with water before you cover with it the most corrugated cardboard. Covering the cardboard with a loose layer of straw reduces evaporation and provides shade. Incubate for several months, periodically watering and checking to see if the mycelium has attached to the cardboard. Once the mycelium covers 25 to 50% of the cardboard, it can be transferred. If it is over-incubated, the mycelium will die back. For this reason, it is best to move the mycelium at the crest of its growth, what I call surfing the mycelial wave. Among the saprophytes listed in this book, mycelial waves may continue for 4 to 2 years before the mycelium consumes the cardboard, is consumed, and/or dies back. Once the underside of the cardboard is colonized, with the growth running through the corrugations, pick up the myceliated cardboard and do either of the following. Place it on top of a new bed of fresh wood chips, mycelial face down. I call these mycelial faces. When placed onto a new substrate, the face becomes a footprint. Use it to make bunker spawn by sandwiching it between 2 burlap sacks filled with wood chips. Use the myceliated cardboard to inoculate more sheets of corrugated cardboard. Simply strip the smooth paper to expose the corrugations, soak the cardboard, and sandwich it between the new cardboard panels. By alternating panels with and without mycelium, you can build a mycelium cardboard tower. Depending upon how thoroughly the mycelium has colonized the paper and the number of walls in the corrugated cardboard, several sheets of mycelium can be delaminated, separated into individual panels, substantially increasing the surface area of mycelium for use as cardboard sheet spawn. When placing cardboard with an exposed mycelial face upon wood chips, make sure the substrate is moist before contact. If living mycelium touches dry wood, the wood will suck the moisture from the mycelium, causing cells' walls to collapse and harming them. If you place the sensitive mycelium onto a wet surface, it can grab onto the surface without having to struggle to manage moisture within its own cells. This strategy allows the mycelium to leap off. Leap off is a term growers use to describe the mycelium's recovery from the shock of inoculation and the ensuing growth surge. Generally, the better the leap off, the better chance the mycelium will win the microbial race. Humans have 5 to 6 layers of skin, whereas the mycelium has just 1 layer. Our thick skin protects our cells from infection and dehydration. Mycelium does not have such protection. The mycelium is surrounded by millions of hungry microbes in the soil, many of which will quickly consume it if it weakens. Young mycelial cell walls are not callous, but sticky. They form a moist membrane through which metabolites are continually secreted. The mycelium is hypersensitive to water loss, and growth stops when it contacts dry surfaces. I try to imagine what the mycelium feels when it is exposed to dry air. I imagine that the feeling is like that of an open wound exposed to blowing wind, a burning pain. In contrast, I think the mycelium must experience a soothing feeling in the rain or fog. Of course, we may not ever know what the mycelium feels, but I do know the end result of mycelium's exposure to dry surfaces or arid air. My recommendation is to transfer mycelium only under moist conditions onto wet materials. By easing the transition and providing healing water, you can help foster cell growth while protecting cell viability. Once you place the cardboard spawn on your new mushroom bed, the mycelium runs downward. If you lay down 2 layers of myceliated cardboard with wood in between, the cardboard barrier helps to prevent organisms from attacking from below or above. In most instances, the lower level of cardboard is placed mycelial face up, with several inches of wood chips piled on top. Cover the top surface with the other myceliated panel face down. Placing 2 layers of mycelial cardboard has been one of my more effective methods for colonizing outdoor habitats. If you are so lucky as to have mushrooms in hand that you want to cultivate, then you have lots of fun making spawn from stem butts. Stem butts have some remarkable, unexpected properties. Stem butt spawn and the one dowel revolution. This method may revolutionize the outdoor cultivation of many mushrooms. Perhaps you collected mushrooms while on a walk, or you have mushrooms fruiting. This is a time to make use of one of nature's most fortuitous fungal opportunities. Natural spawn can be generated from the basal rhizomorphs radiating around the stem's connection to the nurturing mycelium. If you cut the base away from the stem, carefully protecting its rhizomorphs, the tissue stays alive. A few stem butts can inoculate a bed of wood chips or wooden furniture dowels, plugs, or be sandwiched into corrugated cardboard. For years thereafter, you can create dozens of satellite colonies from the stem butts of mushrooms from your mother patches, and each colony can be continuously expanded into constellations of colonies. The effectiveness of the stem butt inoculation technique is a quirk about mushrooms that is unknown to most people, coming as a surprise even to many seasoned mycologists. I discovered this method by accident, and I'm still perplexed by the chi power, the life force energy emanating from the stem base. Practically all connoisseurs of mushrooms trim and discard stem butts before cooking, but surprisingly, with many saprophytic mushrooms, stem butts, but not the areas above the stem base, regrow with astonishing vigor when transplanted into wood chips, cardboard, or wooden dowels. This method of regrowth is another smart evolutionary advantage of the mushroom. Since foraging animals, including humans, prefer to eat the softer flesh of the upper fruiting body rather than the tough, often woody flesh at the stem base. When we throw stem butts away instead of eating them, we inoculate new habitats. To use this method, harvest mushrooms with stem bases intact and ideally with their radiating rhizomorphs, dangling string-like mycelial strands, attached and infused with substrate particles. The rhizomorphs by themselves do not grow as well as they do when attached to a stem butt. The stem butt acts as a nutrient source for the rhizomorphs as they grow rapidly onto new materials. The following 2 methods for growing spawn from stem butts use cardboard and dowels, respectively. Both methods allow growers to expand mycelium. Making cardboard spawn from stem butts. To use stem butts to make cardboard spawn, 1. Gather fresh mushrooms of varieties known to have stem butt regrowing capacity. 2. Using a knife or scissors, clip off the base of the stem just above where it narrows, keeping the root-like rhizomorphs intact and attached to the stem. 3. Soak cardboard in water until it is fully saturated. Tear off sections to expose corrugations. 4. Place 1 stem butt on the cardboard roughly every 16 square inches and sandwich them between panels of corrugation. 5. Soak the stem butts and cardboard in water and place in a cardboard box, trunk, old bathtub, sink, or trough, or simply on the ground and cover with a shallow layer of wood chips. 6. Keep in the shade, with the incubating container on the ground to limit temperature fluctuation. 7. Incubate for 4 to 8 months before transplanting. At this point, you can use each sheet of cardboard as a mycelial footprint to inoculate an outdoor bed, stacked burlap sacks, or straw bales. Or you can place sheets interspersed within a stack of moist newspapers and let them become myceliated over several months. Your imagination is the only limit. However, the mycelium must be moved, transplanted, or it will die. Hence our motto with mycelium, move it or lose it. Making dowel spawn from stem butts. Another permutation of the stem butt technique is to use stem butts to inoculate unsterilized wood dowels. The dowels are first submerged in water for a few days or weeks, drained, and then inoculated with stem butts from fresh mushrooms. In the example depicted in figures 156 to 162, 10,000 dowels half fill a box measuring 12 by 20 by 10 inches and weighing about 20 pounds. Into this box, I place 4 to 8 stem buds, each about 4 inches apart, and push them underneath the surface to a depth of 2 to 4 inches. I covered the inoculated dowels with several layers of exposed corrugated cardboard and placed it in a blackberry patch. After 6 months, I harvested the several large mycelial islands that had grown and used them for inoculating stumps, logs, cardboard, chips, and more dowels. After removing a third of the dowels, I mixed up the remaining ones to help colonization, since at this step in the process, mycelia growth benefits from disturbance. The broken rhizomorphs then regrew with astonishing vigor, soon covering the remaining dowels. These colonized dowels are powerful platforms for launching mycelium onto cardboard, coconut fiber, rope, or burlap bags filled with wood chips, or they could be used to plug logs and stumps, or even to inoculate more dowels. What I like about this method is that the mycelium is acclimated to microbes and is tenacious and feels pure. It produces high-quality spawn that can be expanded many times over. From this stage, you now have many options for traveling down the continuously diverging mycelial path. This method works so well that I call it the one dowel revolution. How much dowel spawn could you make from a single mycelium-covered dowel? Using my experience as a basis, I calculate that the first year you can make 10,000 mycelium-covered dowels from a single stem butt. The second year, the mycelium on each dowel could also be multiplied 1,000 to 10,000 times for a total of 10 to 100 million dowels, weighing about 2,000 to 20,000 pounds after just 2 years. If you use the dowels to inoculate burlap bags filled with wood chips, by the end of the third year, you could have generated enormous mycomass given an inoculation rate of 10 to 100 myceliated dowels for each bag. The number of burlap bags that could be inoculated is mind-boggling, from 100,000 to 1 million. When a burlap bag is laid upon the ground, it covers 2 to 3 square feet, meaning that enough mycelium can be generated to cover 200,000 to 3 million square feet, equivalent to 4.5 to 69 acres of coverage. Cooling bags that fail to show good growth may be necessary. Even if only 25% of the burlap bags fully colonize, a reasonable expectation, this low-tech method can be a dramatic tool for restoring polluted environments and for building life-sustaining soils. If grasses or trees are added into the mix, each bag becomes a pedestal of ecological rebirth. Diverse, complementary biological populations can use each burlap bag as a springboard for growth. For mycoremediation or for growing mushrooms, this technique is revolutionary in its simplicity and large-scale production possibilities. It works remarkably well for generating a large mass of spawn without having to depend upon a spawn laboratory, provided you have patience, have the right strain, and can run with mycelium, planning several years ahead. Mycelial masters could artfully employ this mycelia expansion to craft wide-scale restoration from stem butts. Which mushroom stem butts will regrow? Undoubtedly, hundreds of species fit into this category. My rule of thumb is this: if the mushroom is a saprophyte with rhizomorphs, its stem butt will likely regrow when replanted. However, I encourage you to experiment with the stems of likely and unlikely mushrooms, since new knowledge about species' regrowth abilities could be put to a positive use. The chart on this page lists of some of the more interesting species, all saprophytes, known to regrow. I expect that in the years to come, this list will be greatly expanded as a result of my research and yours. Mycorrhizal species can also be transferred to new habitats using stem buds, a fact that may come as a surprise to some mycorrhizologists. Differing from those of saprophytes, these stem buds, such as those of chanterelles, matsutakes, porcini, and amanita, must be placed directly into the rhizosphere of a young tree or plant host, especially ones lacking mycorrhizae, like many of those coming from indoor nurseries, which are often grown in pasteurized soils. The soil in which the stem is embedded and concentrated with spores, making this region rich in regenerative potential. Whether the mushrooms are saprophytic or mycorrhizal, I encourage you to take the stem buds of mushrooms collected on forays and cast them about your property to create satellite mycological communities. Burlap bag spawn, amassing mycelium or preparations for creating a mycelial tsunami. Burlap bag spawn can be created from inoculations, individually or in combination of dowel spawn, cardboard spawn, stem buds, spore slurries, and pure culture spawn. When spawn is mixed into materials such as wood chips and stuffed into burlap sacks made of jute, hemp, cotton, or wood fibers, a living pod with a purpose is made. Using burlap bag spawn, homeowners can create mycological landscapes in their backyards. On a larger scale, this type of spawn can help repair damage to ecosystems and create buffers between sensitive ecosystems and toxic environments. Immediately upon construction, these spawn bags filter biological or chemical wastes and prevent downstream contamination due to the uncolonized wood chips' extensive surface areas and absorptive abilities. As the mycelium grows, its net-like cells increasingly trap silt and bacteria, and the sweat of the mycelium denatures many toxins, both chemical and biological. Upon colonization, a burlap bag of spawn, now weighing 30 to 60 pounds, becomes a mycofiltration vessel that can also be put to work for habitat restoration or can be staged for further expansion of mycelium into more wood chip filled burlap sacks. These bags, at a glance, resemble sandbags used for preventing swollen rivers from overflowing and hence are sometimes called bunker spawn. In a sense, bunker spawn serves a similar function, preventing shallow sheet flows of contaminated laden sediments and controlling erosion. Vertical walls or horizontal plateaus of myceliated burlap sacks or bunker spawn can be built according to the needed ecological applications and other design criteria. The portability of bunker spawn allows for a variety of applications. I filed a patent on this idea. If uninoculated wood chip filled bags are stacked in contact with myceliated bags in a checkerboard pattern, but no more than 2 bags high, leap off of mycelium onto the virgin bags is rapid and direct. From my experience, stacking the bags 4 or higher results in the growth of competitor fungi. The densest growth takes place near the ground. This simple technology, creating bunker spawn, may be a keystone event, leading to better habitat restoration methods. If the bags are left in place on the ground, oftentimes redworms move in to consume the mycelium, and in turn, a nurturing soil forms, creating a springboard for vast communities of other organisms. The following methods for creating bunker spawn are easy for anyone to put into practice. There are several effective sources of mycelium and suitable materials for creating bunker spawn. Burlap sacks filled with wood chips can be inoculated with mycelium coming from these sources: spores, grain, sawdust, wood chips, or dowels, stem butts, myceliated cardboard sheets. But regardless of the materials you use, the mycelia that result have one thing in common: a preference for fabrics. The architecture of fabric, specifically that of burlap bags, is well suited to rapid colonization by mycelium. The mycelium, being a fabric itself, recognizes this structure and grows through it with a surprising velocity. The breathability and biodegradability of the fabric are essential for success. This biodegradable membrane allows for rapid colonization by the mycelium, encompassing its contents like a living pillowcase. Inside the fabric membrane, the mycelium grows outward as well as inward. This mycelial conversion grips the substrate to the disadvantage of competitor fungi, which failed to compete with the ever-advancing avalanche of mushroom mycelium. In effect, the surface fabric of burlap sacks becomes the contact point for the mycelium to leap off into the contents. By first inoculating the sac, we allow the mycelium to run quickly through the membrane, and encase the contents of the bag with a sheath of mycelium on burlap. Since the fabric's tensile strength deteriorates as a result, the bag will degrade in about 6 to 12 months, falling apart and spilling its mycelial package. Here are some of the diverse ways you launch mycelium into burlap sacks filled with wood chips. Some methods will be more appropriate than others depending upon your access to natural or commercial sources of spawn mushrooms. Spore prints and spore mass slurries for inoculations of burlap sacks. Create a spore mass slurry using 1 gram of spores from, for instance, 6 oyster mushrooms, 4 to 6 inches in diameter per gallon. The typical size of burlap sacks that have worked well range from 12 by 24 inches to 18 by 30 inches. Larger bags become unwieldy due to their weight. This method works best if the burlap is hot water, pasteurized, sterilized, or chemically treated using hydrogen peroxide to reduce spores of competitor fungi in the fabric. If burlap is soaked using hydrogen peroxide, make sure it's thoroughly rinsed with clean water after treatment, lest the residual peroxide harm your implanted mushroom spores. Once spores germinate into mycelium, however, residual peroxide levels are usually not detrimental to the implanted fungus, as the mycelium's digestive enzymes contain natural peroxides. Once the bags have been treated and inoculated with the spore mass slurry, fill them with wood chips and stack them into a wall 3 tiers high. After the bags are saturated with the spore mass slurry, or after adding water once prints have been made upon them, roll up the bags and stuff them into the cardboard mailing tubes. Incubate for a few months outdoors and then remove the rolled burlap from the mailing tubes. Oyster mushroom mycelium will produce whitish zones of growth in 2 months to 6 months after contact. Select the best colonized bags. Unroll the bags with the best myceliation, stuff them with wood chips, tie them closed, and stack them 3 high on a pallet outdoors. Try to get the mycelial faces to contact uncolonized areas of the bag stacked below or above them. This method, although it can work, is not nearly as predictable or successful as the methods described below. Caution: when using spores or when using these low-tech methods, wear a filter mask and avoid black, yellow, or brightly colored molds. Some of these can be allergenic or even pathogenic. Use of commercial spawn grain, sawdust, wood chips, or dowel for the inoculation of burlap sacks. Pure culture spawn is available from a number of commercial laboratories. The success of using pure culture spawn to inoculate raw wood or straw is limited because of the competition from the millions of spores already resident in that material. Having been raised in a sterile laboratory and never before exposed to competing microbes, the mycelium's immune system must work to overcome its competitors. To give pure culture spawn a chance to devise defensive strategies in advance, a few days before use, introduce a small sample, a few grams, for example, of the raw substrate to the spawn, 5 pounds, for example. In effect, you are purposefully contaminating the pure culture spawn with a sample of the microbially rich habitat in which it is destined to reside. As is the case with vaccinations and homeopathy, exposure at low levels fortifies the immune system, preparing it for its impending contact with the many populations resident in the natural world. Typically, pure culture spawn added to a grain substrate in burlap bags does not work well unless the substrate has had its population of undesirables knocked down using heat treatment, sterilization, or pasteurization, hydrogen peroxide, or any means that renders competitor spores ineffective without damaging the receptivity of the substrate. Pure culture spawn added to sawdust substrate tends to work better in many situations, but leap off the ability of the mycelium to recover from the shock of being introduced to a new substrate is considerably slower. A clever method we have developed for growing both grain and sawdust bunker spawn is what I call cold incubation, the inoculation of raw substrates during cold outdoor temperatures, 35 to 45 degrees Fahrenheit. At these temperatures, most major competitors are dormant or have such a slow rate of growth that they are unable to compete with your introduced spawn, which must be a cold weather variety in order for this strategy to work. After several months, as temperatures rise with the advent of spring, the mycelium zooms to the forefront ahead of competing microbes and is better fortified to defend itself from microbial attack. This procedure of using spawn in combination with a cold incubation strategy is useful for myco-restoration strategies. One variation of this strategy for creating bunker spawn stands out. inoculating bags of wood chip-filled burlap sacks with wooden dowels, made either from pure culture spawn or from stem butts. Surprisingly, each dowel or plug by itself has enough nutritional mass to allow mycelium to project deeply into the surrounding substrate. Dowel spawn, traditionally used for inoculating logs, can also be used for creating outdoor beds. Cold incubation works particularly well with dowel inoculations of temperate mushroom species of burlap sacks filled with wood chips. Experimenting with combinations of strains and materials will give you valuable insights regarding the best methods. Stem butt inoculations of burlap sacks. Stem butts can be deployed in either of two ways: interspersing them throughout the wood chips in a burlap bag, or sandwiching the stem butts between two bags after they've been filled with wood chips. For both of these methods, the workable rate of inoculation is 5 to 10 butts per 40-pound bag of wood chips. If you want to create many sheets of mycelium from stem butts, first soak similar sizes of flattened burlap in torn, corrugated cardboard in cold, clean water. Place at least 2 stem butts per square foot onto the grooves of the corrugated cardboard. Cover with a flattened burlap sack, add another layer of moistened corrugated cardboard, add more stem butts, and so on. This lasagna building approach will result in a tower of mycelium, which should be stored outdoors on a wooden pallet, slightly above ground, in a cool, shady place. This mycelial tower needs to be broken down, separating the cardboard from the burlap within 3 to 6 months, since the bags will fall apart if the tower is over-incubated. Optimal timing balances colonization by the mycelium, tensile strength of the bags and cardboard, and separability of the layers. After the tower is taken down, the mycelial bags can be filled with substrate, while the cardboard can be inserted between newly filled bags of wood chips. Cardboard spawn inoculation of burlap sacks. Cardboard spawn, which can be created using spores, spawn, stem butts, or transplanted face or footprints from overlaying mycelium, is an effective tool for inoculating the interface between two burlap sacks. The sacks are first filled with wood chips and soaked with water, and then, while they're still moist, the cardboard is delaminated to expose the mycelium. Immediately after exposure, the mycelial face is placed directly on the soaked burlap. Another soaked burlap bag filled with wood chips is laid on top. Together, the bags are covered with either loose cardboard or shade cloth and incubated outdoors until colonization is substantial. Don't wait for 100% colonization, since this spawn should be moved at the peak of its surging growth. By inoculating a population of bags and spot checking their growth, you can get a good sense of whether the mycelium's growth rate is climaxing or descending. It's always best to run with mycelium when its pace is the briskest, projecting it at its optimum rate of growth. Once it peaks, vigor declines, and the mycelium loses its momentum. I follow this rule at each step in the expansion of mycelium. Bunker spawn for habitat restoration. Bunker spawn is an ideal tool for habitat restoration. It can be used on a commercial scale and in many different ways. Bunker spawn can be placed on the peripheries of watersheds to protect them from the surface flow of contaminants. Bunker spawn can also filter contaminants emanating from chicken, pig, and cow farms, in the process protecting downstream environments from bacteria, viruses, protozoa, pesticides, nitrates, and many other pollutants, including the so-called inert ingredients commonly used in fertilizers. Adding bunker spawn to riparian buffers creates a mycofiltration layer, even on the first day of application. The wood chips, with their large surface area, catch substantial colonies of bacteria. The benefits of inoculating wood chips are that you can be selective about the fungi you grow, and that colonization is far faster than it is in the wild. Uninoculated fresh wood chips are vulnerable to spore fall, wild spores raining down from the air and coming up from the ground. Spore fall typically produces island colonies of several competing species, usually 2 years after wood chips have been manufactured. Essentially, the window of opportunity for getting a species you desire to grow on recently chopped, undiseased wood chips usually closes in 2 years, when native fungi surge and dominate. In contrast, inoculated wood chips colonize with mycelium in 6 to 12 months, and the mycelium, secreting antibiotics and free radical enzymes, destroys many airborne and embedded spores of competing fungi. Selecting mushroom species with unique filtration or remediation properties allows the landowner to customize a mycological response specific to the environmental threat. When creating bunker spawn, selecting native mushrooms is recommended over importing foreign species. For instance, I recommend this method for growing garden giants, clustered wood lovers, turkey tails, cauliflowers, and some woodland agaricus mushrooms. No doubt, dozens if not hundreds of species of mushroom could be used in bunker spawn production. Whether or not the substrate is inoculated with grain spawn, plug spawn, stem buds, or laboratory-grown sawdust or chip spawn, I also recommend the bunker spawn method for getting extra crops of shiitake, turkey tail, or reishi mushrooms by recycling spent substrate from a gourmet mushroom farm. If not for the extra mushrooms that can be produced, the best use of this form of mycological currency may be for their pollution-destroying enzymes, placing them in key locations in and around toxic waste sites. If bunker spawn is being incubated in staging area before placement, the burlap sacks should be incubated near to but off of the ground, for instance, on wooden shipping pallets or sheets of cardboard. If incubation is to occur on the site where you want your mycelium to reside, then bunker spawn bags can be laid side by side to create 4 to 6 inch deep contiguous layer of wood chips directly upon bare ground. I prefer to place the mycelium face down rather than up in the air. The mycelium digests the wood chips and the bag over time, beginning the process of soil building. Not every bag of bunker spawn will colonize. Often, spots of dark green and black mold can appear, but they are weak competitors to those mushroom species that project ropey rhizomorphs. The art of using the bunker spawn method is in knowing which bunker bags of mycelium to expand and when. Sacs that are poorly colonized should be removed. On the other hand, the sacs that become fused together by thousands of interconnecting rhizomorphs have enough chi power to leap off into fresh wood chips, allowing deep penetration and overcoming ecological barriers that weaker mycelium could not. These are the bunker spawn bags I select for expansion. One strange twist in this method is that often the mycelium from the burlap spaces penetrates more effectively to the depths than does the mycelium from within the bags. I think the mycelium mat that forms in the burlap surface becomes a platform of strength, a membranous sheet of cells that helps the mycelium project its tendrils deep into the depths of the internal wood chips. Once a naturalized bag of bunker spawn has matured, in many cases becoming snow white in color, this spawn can inoculate more wood chip-filled burlap sacks for maximum mycelium mileage or be mixed into wood chip beds to create mycological landscapes. Bunker spawn generated by any method of inoculation, stem bud, pure spawn, native mycelium, cardboard, sheet spawn, or spore mass slurry creates a mycelial footprint of substantial mass. The methods described here could generate hundreds of tons of mycelium. The sacks are easily used in conjunction with commercial filling systems, chippers, and bark blowers. Mulching equipment already in use by restoration and landscape companies would require only simple modifications in order to serve mycological needs. Naturalized bunker spawn works superbly and is less expensive than pure culture spawn. This simple technique may be the bridge of knowledge that so many myco-restoration and bioremediation projects have needed in order to achieve success. Combination methods for creating bunker spawn. Mushrooms are often mysterious in their ways. Sometimes they inexplicably fail to grow. If the substrate is suitable and none of above methods have worked, then I recommend that combinations of the previously mentioned strategies be used. Combining spores, stem buds, and mycelium to create a complex inoculum often leads to development that surges past barriers that would ordinarily stop the mycelium if only one method was used. The bottom line, if you want the best chance of culturing your mycelium, then inoculate the burlap sacks using 3 methods: spore printing, stem bud inoculation, and mycelium transplantation. If the conditions are right, then the surge of regrowth is nearly unstoppable. Preferred paths of expansion for bunker spawn. Now that I have described several methods of getting the mycelium to run using bunker spawn, what paths are likely to lead to the greatest success? Methods that are most effective in the Pacific Northwest may not work as well in Thailand. Nevertheless, the principles are the same. It's the technique that may differ. As a cultivator, you will face critical decisions as you follow the path toward mycelium expansion. The good news is that several paths lead to success. Which one is best for you depends upon your experiences. Here are 3 paths that I prefer in my bioregion. Pure culture naturalized plug spawn mixed into fresh, raw wood chips in burlap bags, 100 to 200 plugs per sack. Sawdust spawn or spawn from a recycled mushroom kit stuffed into wood chip filled burlap sacks. Stem butts used in 2 ways. A. To make wood dowel spawn, which is in turn mixed into wood chips placed in burlap sacks. And B. To make wood dowel spawn, which is in turn mixed into wood chips to make a mushroom garden as shown in figures 157 to 159. The first uses laboratory-grown spawn, preferably acclimated. The second takes advantage of natural spawn. The third uses stem butts as a source for mycelium. All 3 can create bunker spawn, which can subsequently be expanded. However, the third method is particularly favored, since once the mycelium naturalizes, meaning that its immune system adapts to the complex microbial environment in the wild, it really runs and it is aggressive. Pure culture spawn and recycled mushroom kits, on the other hand, must overcome adverse microbial hurdles before the mycelium can move. Subsets of this second generation naturalized spawn can be selectively expanded by at least 2 orders of magnitude. In effect, each bag of bunker spawn can generate 10 to 100 more bags. Once mushrooms begin fruiting, stem butts from the new crops can inoculate dowels or corrugated cardboard panels, thus renewing the cycle of expansion. Growers who can successfully expand the mycelium by incorporating a variety of propagation techniques heighten the art of cultivation. Like an orchestra conductor, a good grower conducts a strategy that makes the best use of the instruments in the theater and is sensitive to peculiarities of the players. Alternative materials for growing mycelium and mushrooms. Most new cultivators are astonished, as I was, by the variety of waste products mushrooms can decompose. For example, most of the major debris that comes from farming or forestry can be used in some fashion by mycologists. Many other materials listed below can also be used. Mycelium grows best on most of these materials when they have been broken into smaller pieces, 1/4 of an inch to 2 inches, and are 60 to 80% saturated with water. Mixing fine and coarse materials together often creates a friendly matrix for the mycelium, since it loves complex mixtures with fractal-like microstructures. However, with most of the species in this book, consistently using fresh material that is known to be hospitable to your mycelium can make the learning curve less steep. Many of these materials are discussed in more detail in Growing Gourmet and Medicinal Mushrooms and the Mushroom Cultivator. The excellent website www.mushworld.com also provides extensive lists and details recent cultivators' successes. Mushworld has published 2 excellent mushroom growers' handbooks, Oyster Mushroom Cultivation, Mushworld 2004, and Shiitake Mushroom Cultivation, Mushworld 2005, distributed for free to developing countries in an effort to help fight poverty. The list of alternative materials below, compiled from the collective experiences of cultivators worldwide, will expand as cultivators like you continue to experiment. These materials can be processed using the methods described in this book for making spawn, expanding natural mycelium, and creating mycelial mats. Bamboo, brewery waste, cacao shells, cacti, coconut, coffee bean grounds, hulls and debris, corn cobs and corn stalks, cotton and cotton waste, fabrics, garden waste, grass clippings and yard debris, hair, hemp, leaves, manure, nut casings and seed hulls, oils, vegetable and petroleum, paper products, newspaper, cardboard, money and books, soybean roughage, straws, wheat, rye, rice, oats, barley, etc., sugarcane and sugarcane tea, tea waste, leaves and trimmings, textiles, tobacco and tobacco stalks, trees, shrubs, brush, and wooden construction waste. Water hyacinth. If you are uncertain which material to choose for running mycelium, choose a few that are readily available and run a preference test by placing mycelium on each one. By inoculating several materials on the same day, you start a mycelial race. The materials most quickly colonized, as a general rule, are usually the best. Experimentation using mini trials will give you ideas that can make larger projects less tedious and more likely to succeed. Have fun with your fungus.